This video is going to look at section 6.4, which will continue our look at cellular respiration. And so far what we've looked at is the phase one of cellular respiration, which is just simply glycolysis. And that takes place in the cytoplasm of the cell. And it does not require any oxygen. It does require 2 ATPs, which will activate the glucose. And when that takes place, then the glucose gets split into two 3-carbon molecules, which are known as pyruvate. And it does yield in the end, you do get 2 ATP. There's a net gain of 2 ATP from that process. Again, it doesn't require oxygen, and if there is no oxygen present, then what will happen is we get fermentation, which is what we've looked at already as well. So now what we'll look at are the final 3 phases of the breakdown of glucose in the presence of oxygen. So when oxygen is present, then everything then moves inside the mitochondria. So this is phase 2 now, and so this is what's referred to as the preparatory or the prep reaction. So when oxygen is present, then the final reactions of cellular respiration occur. So phase 2 then is preparatory reaction, phase 3 is a citric acid cycle, and then phase 4 is electron transport chain reactions. So this is phase 2, and what just simply happens here is that the pyruvate then is oxidized to acetyl coenzyme A, which will then enter the citric acid cycle. So again, actually oxygen is not required for this. However, in order to enter the mitochondrion, then oxygen does need to be present. So in the presence of oxygen, then things move inside the mitochondria, and then we get the prep reaction, which is phase 2. And then to continue on with that, now we have phase 3. This is the citric acid cycle. So this also occurs within the matrix of the mitochondrion, and again, it's a cyclical pathway, and it converts the acetyl groups to carbon dioxide. So in this phase, ATP, NADH, and FADH2 are produced. And so here's a summary of this cyclical type of pathway, and you can see that the pyruvate now has become a 2-carbon acetyl group, which combines with a 4-carbon molecule of the citric acid cycle, and then at various phases or various stages, I guess I should say, CO2 is given off, and oxidation reactions produces NADH plus H plus, and ATP is produced, and FADH2 also is produced from this cycle. At the end of the cycle, then the 4-carbon molecule reemerges, and then it is able to once again accept another acetyl coenzyme A, which then again causes the cycle to take place. So this is a very good summary of all the things that, well, most of the things that take place here. And so this is again, this is phase 3, and this is the citric acid cycle. So what we put into this, we put 2 acetyl groups, 6 NAD plus, 2 FAD, and 2 ADP plus 2 phosphate groups. And so what we get out of this is the carbon dioxide is given off, 4 molecules of that. It yields 6 NADH plus H plus and 2 FADH2, and 2 ATP are yielded in this particular cycle. So again, this is phase 3, and this is the citric acid cycle. So that brings us to our final phase, and this is the electron transport, ATP synthesis. This now takes place within the cristae of the mitochondria. And what happens is that electrons are passed along a series of carriers, and high energy electrons enter the system, and obviously then low energy electrons exit the system, and the difference in energy then is what's captured by the system. So NADH plus H plus becomes NAD plus, FADH2 becomes FAD plus, and then energy is captured in the form of a hydrogen ion gradient. So O2 receives the electron, the low energy electron that exits, or electrons, I guess, that exit, and then react with H2 to form H2O. And again, there's the diagram of that taking place there. So here is the reaction pathway here, and you can see at each of the stages where different things are produced, where we produce ATP, FAD plus 2H plus, and NAD plus 2H plus as well. So there's a good summary then. So this is the, again, phase 4 electron transport chain, and this takes place within the cristae of the mitochondria now. So the components of the electron transport chain, they have a specific arrangement in the cristae. So hydrogen ions are pumped from the matrix to the intermembrane space, and this produces an unequal distribution of hydrogen ions called an electrochemical gradient. And so the H pluses, they move back from the intermembrane space to the matrix by passing through the ATP synthase complex. And so this causes the enzyme complex to produce ATP from ADP and phosphate. So ATP synthesis here then is said to occur by chemiosmosis. And here's a good diagram of how this takes place. So this is within the cristae of the mitochondria, and so the intermembrane space is here, and you can see there's a higher concentration of hydrogen ions there, and so in order for the concentration to become restored, then the hydrogen ions have to move through the ATP synthase complex, and when that takes place, then ATP is produced because of that. So this is where we get our greatest energy yield, is in this last phase. And so the maximum ATP yield from the complete oxidation of glucose can be calculated in this manner. So from the first phase, glycolysis, that yields 2 ATP. Remember, this is from one molecule of glucose. So the first phase, glycolysis, we get 2 ATP. The citric acid cycle yields 2 more ATP, and we're now within the matrix of the mitochondrion. And then we have the electron transport chain and chemiosmosis, and this will yield 26 to 28 ATP. So experimental observations will show that we get 2 to 3 ATP per NADH in the electron transport chain, and 1 to 2 ATP per FADH2 in the electron transport chain. So in many cells, NADH produced in the cytoplasm by glycolysis requires ATP for transport into the mitochondria. So here is again the different phases and where the energy is yielded in each one of those, and where it actually comes from. And so you can see that again, it's the very last phase, the electron transport chain, where the majority of the energy is captured from this entire process. So we could also determine the efficiency of cellular respiration. And so it has been determined that the difference in energy between the reactants, glucose and O2, and the products, CO2 and H2O, there's a difference of about 686 kilocalories of energy. So the breaking of 30 phosphate bonds and the conversion of 30 ATP to 30 ADP plus 30 phosphates, that will yield 219 kilocalories. So therefore, just like any system that wants to or that extracts energy from anywhere, it's never a perfect system. You will never yield 100% of the energy that's available. And so once again, in this case, it's about 32% efficient. And so we will get about 32% of the available energy from glucose converted to ATP. And the remaining energy then, that just simply dissipates as heat. And that would be true of any system that where you want to extract some energy. It will never be a perfectly efficient system. You will never achieve 100% energy efficiency in any system. There will always be some heat given off, and in this case, we are able to capture about 32% of the energy that's completely available. So that will end our look at cellular respiration. And so once again, here's the key terms and the key concepts, and a summary of what we've looked at here, and once again the summary of the key concepts. So we will leave this topic now, and we'll move on to other things.